Technical Veterinary Learning TVL channel, founded and produced by Professor Ahmed Mamdouh El Sharif, Professor of Epidemiology and Infectious Diseases of Animals, the ex-head of Department of Veterinary Medicine, the ex-Vice Dean of Faculty of Veterinary Medicine, Bani Suef University. Laboratory diagnosis of skin diseases, mange and ringworm. Accurate diagnosis of different infectious diseases of animals is the true base of effective control to them. Diagnosis done through use of definite knowledge from one or more basic sources of information, namely history of the disease, epidemiology of the disease, clinical signs of affected animals or herds, post-mortem findings, and results of different laboratory tests. Examination of diagnostic materials is an important function of veterinary laboratories that help different veterinarian practitioners in effective treatment and for application of different control measures against resurrection of this trouble again. Skin scrapings and hairs are needed for examination for diagnosis of mange and or ringworm. Skin scraping sample. Diseased cutaneous areas examined carefully before collection of samples. These areas then wetted with mineral oil to prevent distribution of suspected scrapings around during collection. Skin scrapings collected from edges of suspected areas after picking up of a cutaneous fold tightly between thumb and forefinger. Skin scrapings which adhere to the blade should be transferred to any of the following, namely a clean dry petri dish, test tube, plastic sheet, paper envelope for culturing and or microscopical examination. Hair samples. Hair samples. Hair specimens were collected from the edge of suspected area by usage of suitable forceps. These samples were used for culturing and or microscopical examination of these hair shafts. This picture showing how to collect skin scrapings at neck area of an infected cattle. Collection of skin scrapings from a neck by usage of blunted scalpel appear in this figure. Here the shape of a skin scraping sample after collection. Examination of skin scrapings for diagnosis of mange disease. Skin scrapings are kept in 10% of potassium hydroxide or 10% of sodium hydroxide for 2 to 4 hours and may be gentle heating applied during this process. Then centrifugation at 3000 revolution per minute for 10 minutes was applied with discard to the supernatant. Then a drop of sediment is examined under low power of the microscope for detection of different types of mites as shown in the following figures. This figure showed odd lesions of mange in a cattle where scabby large areas of the skin, wrinkled, found around eyes, at the jaw, and all over the neck. Distributed lesions of mange in a cattle at the whole neck area and at the beginning of the dewlap. Distributed lesions of mange in a cattle at the whole tail, at the whole buttock area. This is the figure of Psoroptes species of mange. A figure of Coreoptes species. A figure of Psoroptes species. A figure of Sarcoptes scapy species. A figure of demodectic mange species. The shape of Sarcoptes scapii, both adult and eggs. Adult of Sarcoptes scapii. Examination of skin scrapings and hairs for diagnosis of ringworm disease. Ringworm in the face of a cattle, taking ring shape with allopatic area and skin scrapings. This is part of the face, the side long of the cheeks and under the ear, and also around the eye. Ringworm in a cattle, around the nose, around the eye, around the ear, and in the front head. An old lesion of ringworm in a cattle, in the neck, in the dewlap, in the face, around the eye, along the side of the jaw, and around the ear. Skin scrapings and hairs were placed on slides with 10% of potassium hydroxide or 10% of sodium hydroxide, then covered with cover slips, and gentle heating was done. Then examination under low and high lenses was done for the latter slide. The presence of costive hyphae and spores were seen between skin scrapings as well as inside and outside infected hairs as shown in the following figures. This is the shape of endothorax, means presence of arthrospores of the fungi inside affected hair. This is the shape of endothorax, means presence of arthrospores of the fungi inside affected hair. This figure showing the ectothorax means presence of arthrospores of the fungi outside the affected hair. A diagrammatic figure showing the ectothorax and endothorax. By means presence of arthrospores outside and inside of affected hair. This is a figure of microscopical appearance of trichophyton. Golden points for sampling. These golden points have to be followed for a good sample collection. The first golden point, veterinarian clinician should remember that errors could occur during taking of any specimens, so they have to cleanse two representative sample truly. The second golden point: errors in collection of specimens could affect on important results, as in case of parasitological detection of eggs, detection and isolation of mycobacterium organisms, contamination of milk samples with fecal matter. The third golden point: delaying in transferring of samples to the laboratory may result in error, as WPCs are reduced by 10% within 24 hours from taking of their sample. Lab diagnosis will be continued. Please subscribe the channel to follow the newest videos about veterinary medicine.